that you can't see. It's underground, and here on the right, let's see, yeah, can you see the pointer? I didn't ask them for, they had another one. So this is the mycelium. This is the part that's underground, and it is the vegetative part of the fungus organism. And it gathers nutrients in different manners according to what kind of fungus it is, and gathers the energy to put up these fruiting bodies whose function is to drop the spores that it produces so it can begin a cycle of renewal and the spores grow into more vegetative fungal masses. And here's kind of a simple description of what a mushroom is. It compares the mushroom to the apple. So the apple is the fruit of the tree. It produces seeds. The mushroom is the fruit of the fungal organism, and it produces spores. This is kind of a diagram of how a mushroom matures. So it's hard to, it's a cycle, so where do you start? Okay, well, I guess we can start with the spore, and the spore grows into this little rudimentary mycelium, which grows further. Eventually, the mycelium puts up these fruiting bodies, which start out, this is an amanita. Oops, where did I go here? This is an amanita, so it's kind of the most typical mushroom. There's the cursor. It starts out looking like a little egg, and as it grows, the tissue that covers it breaks open, and finally turns into an amanita, which has tissue here, which we call an annulus, and tissue here at the bottom that we call a vulva. And you'll see that later. We have some. This shows you how the spores are actually formed. So here's your mushroom, and this is a cross section here showing you the tissue of the cap. And here are the gills, and the gills produce the spores. Using my cursor. Oh, there it is. Sorry, this is technical difficulties. And then over here is a microscopic view of the tissue on the gills, and these big cells here are called basidiophores, and they produce the spores. The technical name is basidius spore. This is what the spores actually look like if you're looking under a 1,000-power microscope. Let's talk a little bit about how the fungal organism gets the energy to make those mushrooms. Well, there are different ways, and some of them are actually parasitic, and this is one that is, this is Armillaria melia, honey mushroom. It's an extremely variable fungus. It has many different forms, and someday they'll figure out that they're all different species, I'm sure. But you often see it growing at the base of a tree that is dying, or sometimes they're not dying yet. Unfortunately, I just discovered some at the base of my great big huge spruce tree at home. But they do not bode well for the plant. And this is a cordyceps. Cordyceps is a very interesting fungus. It is parasitic on, in our area, truffles, and also on insect larvae. And I think this is Cordyceps capitata, but there are other forms of cordyceps also. Then there are mushrooms whose job is decay. They're the cleaner uppers of our earth, and these little guys are digesting these needles, fir needles, I think. And these are other decomposers. There's a lot of these out right now. This is a hygrophorus, and it is a beautiful fungus, and you find it, especially in redwood forest, there's lots of needles and duff for it to decompose. And this is a wood rotter. This is Fomitarius. It's a conch, hard, woody thing that is perennial. It just keeps growing. and its mycelium are decomposing that great big log that it's growing on. And you often see smaller forms of this, they call them conchs, and they're growing off of trees that are usually dead or dying. This is a beautiful Pholiota. And Pholiota is a group that all grow on wood, and they also decompose wood. Now, most of the mushrooms that we see are not any of those. I mean, we do see those, but the majority of the fungi that we notice and are aware of are different than all of those. They are mycorrhizal, so they grow in association with the roots of trees. It's a mutualistic relationship. The fungus gets carbohydrates from the tree. Because the fungus isn't green, it doesn't have chlorophyll, it can't get energy from the sun to produce its own sugars and carbohydrates. So it gets them from the tree. And the tree gets from the fungus minerals like phosphorus and nitrogen, and also especially moisture. You can see all of this fine white stuff is the mycelium of the fungus. The darker white stuff here is little root hairs, and then these are our main roots. So you can see how greatly it increases the area of absorption for the tree to gather water. So most of the mushrooms we see in our forests are mycorrhizal. Boletes are mycorrhizal. This is Boletus smithii. And this is Cortinarius, now called smithii. I didn't realize they were going to both end up being smithii because they used to have different names. It's mycorrhizal with pine. Okay, let's talk a little bit now about how we identify these mushrooms. So the most important thing, the first thing you start with, is what is the hymenium? Hymenium is the spore-bearing surface. And you can see on these that they have very sharp edge, very sharply defined gills. So these are gilled mushrooms. So that's the first thing you look at is turn the thing over and what has it got underneath? Well, this one has gills. And here's a close-up so you can really see how sharp and knife-like these gills are. And then there are the ones that have tubes. So instead of gills, the spores are manufactured in these tubes, and in the section where it's on its side, you can see that you can just peel that spore-bearing layer off, and those are the whole family Boletaceae has that method of spore production. And this is a diagram that shows you how this is the tube. So the inner lining of those tubes are covered with the spore-bearing cells. The cells are produced there, and then they are discharged actively down through those tubes. And this is a cross-section of a tube showing you the spore bearing cells and the spores themselves. So that's the boletes, or the pored mushrooms. They both work the same way. And this is Albatrellus fletii. It's a polypore, and it also has little, if they're not tubes, they're just little pores. They're very shallow, and they can be confused with tubes, except with these, they're tightly attached to the cap, and you can't peel them off. These mushrooms usually are also not real fleshy like the boletes. And then there are the chanterelles, which have ridges instead of gills. And this is one of the first things that most people learn about mushrooms because they're interested in chanterelles, is how to tell a chanterelle from a false chanterelle. And this is how you do it. These have these blunt edges, not sharp edges. Here's another one, close up, and you can see that that has a rounded edge, not a sharp edge. And this is another type of, in the member of the Canthrollaceae, this is Gomphus clavatus, which has a common name. Most mushrooms don't have common names. And so one of the big problems for people in learning about mushrooms is that there aren't common English names. There's all these Latin names. But this one has a common name, and it is pig's ears. And don't ask me why, I don't know. But It has more wrinkles than ridges. And this is Craterellus cornucopioides, and it doesn't have hardly anything at all, just a few little wrinkles, and mostly it's just smooth. And these are found up in our tan oak forests here, and they're considered a choice edible. They're called black trumpets. And then there are tooth fungi, and you can see in the bottom picture the detail. They have little teeth. They don't have no gills, no ridges, no wrinkles, no tubes, but they have these little teeth. And then there are some, like the clavaria, that don't have really any structure at all. It's just the whole surface is a spore-bearing surface, completely smooth. This is another example. This is Spiraceous radicata, which grows on wood, and it has just completely smooth spore-bearing surface also. It's called cauliflower mushroom. And the Ramarias, the coral fungi, are also just completely smooth spore-bearing surface. So some of the features that we need to recognize after we have determined what kind of spore-bearing surface it has is spore color. And mushrooms will drop their spores in such a way that you can actually take a spore print. This one was done in a petri dish, but you can do this at home with a piece of paper and a custard cup or a coffee cup that you put over the top of it. Moisten your hands, run your moist hands around the inside of the cup so there's moisture there. Put the cup over the mushroom that's on the paper and you'll get a spore print. Usually, unless the mushroom is really, really old or it's too young to sporulate, but usually you'll get a spore print.
It's because it's dark that makes it, is that the coffee cup to make it dark, or?
No, the coffee cup is to keep, good question. The coffee cup is to keep little breezes from blowing the spores around, so they'll actually give you a print like that. This is a black spored mushroom, Phenilus. Spore prints can be really beautiful too. Come on in. Spore prints can be really beautiful. They, you'll get a beautiful pattern of the gills. I know people who have made greeting cards out of spore prints. They're really kind of an art form. You can also get spore prints in other ways. This mushroom, Pleurotus striatus, the oyster mushroom, has left a spore print right down here on the wood that it's growing on. So as you're looking at a mushroom in nature, you often can see the spore color just by looking underneath the cap. Sometimes, this is a entoloma. They have pink spores. So this is, we would call it more like flesh color, but the technical term is pink. These are pink spores, and sometimes they catch right on the stipe of the mushroom. And in mycology, we refer to the stalk as a stipe, and I will probably use those words interchangeably. So this is pink spores that have caught at the apex of the stipe. And sometimes you can just look in a clump of mushrooms at the mushrooms that are below, and you can see these are brown spored. This is Foliota terrestris, I think, and it's the mushrooms above have dropped brown spores on the mushrooms below. There you go, you found a place. This is a Cortinarius. They have rusty brown spores, and also on this one, the spores have caught on tissue at the apex of the stipe. And this is a Pseudocoprinus disseminatus. They come in a big group, and they have black spores, and you can see that the upper ones have dropped the black spores on the lower ones. So I don't like this slide very well, but it's the only one I have. And so it shows you the different spore colors you can get. So on the left you have purple-brown, and that's the color that the agaricus mushrooms, the store-bought mushroom, have purple-brown spores. They're just almost black, but they're actually purple-brown. And then there are black spores, and there are various brown spores. Then there's the rust-brown spores. Then there are the pink to flesh brown spores and white and light spores. So those are all representations of the different shades of spore color that you find in mushrooms. Okay, so we talked about spore color, and now the presence or absence of a vulva, the remnant of a universal veil, which identifies the amanitas. So this is a very important feature to know, since we know that amanitas are generally poisonous. So on the left, you have a little button of an amanita. It's completely closed up, and this warty material that we see is called a universal veil. And as the mushroom grows, it expands, and the veil begins to break up. The tissue that's covering it begins to break up. And then, as it grows more, the tissue breaks up even more, and how it leaves remnants on the cap is one of the ways you identify the different amanitas. So this one has little warts on the cap. That's Amanita muscaria, and that's typical of it. Here's another picture of an older Amanita muscaria, and you can see that this tissue here is called the partial veil. It covered just the inside of the cap to protect the young gills. And so as it falls open, it may do one of many things. It may leave a skirt-like veil on the stalk. It may go away entirely. It may leave just little remnants that you really have to look for. And then the vulva, the universal veil, is now, there's remnants of it down here, and these remnants are called the vulva. And then here you have the little warts that are left on the cap. So that's Amanita muscaria, and they come in red, yellow, and white. This is Amanita pacicolia, and it has a saccate vulva. So you can see there's the cap. It hasn't risen up yet, but it's sitting in this big sac-like piece of vulva tissue. Amanita braconii, and that last one didn't have a remnant on the cap, so all of the tissue is in the vulva. None of it is on the cap. This one has just a patch on the cap, not little warts, and it also is sitting in a big sac-like vulva. This one has big patches on the cap. It doesn't have little warts, it has these big patches. This is Amanita pantherina, and it has a very well-defined rimmed bulb. You can see there's just a real definite edge to that vulva. This is Amanita lanii. It grows in our tan oaks here. It's a really beautiful mushroom, and it has a big patch on the cap also. And it also has a big saccate vulva. It's hard to see here because it's under the leaves. And this is a nice picture showing a button that hasn't opened yet and how it's covered by that tissue. This is the one Amanita that people eat, or that most people eat. There are some people that would eat anything, but this is considered an edible. But I really do not recommend eating Amanitas until you really, really know what you're doing. And while we're on the topic of Amanitas, I will say that there's a mushroom that looks quite a bit like that, and that is Amanita phalloides, the death cap. And we do not have Amanita phalloides here in Humboldt County. 30 years ago, when I started learning mushrooms, Dr. Largent said, but it's coming, it's coming. Well, it hasn't come yet, but we still say that you need to watch out for it because someday it may very well show up here. It's not a native mushroom. It was introduced, came in from Europe on the root of some imported tree and spread from New York, I believe, all the way west. And they have it in Sonoma County, but we don't have it here.
How would we get it here? Just a spore?
It would come on the spore. It would be transported somehow. It would come in on trees that were, landscaping plants that were brought here, for example, just the way it got to New York. But there seems to be something about our climate or our soils or something that it doesn't particularly like because it hasn't showed up here. But go ahead.
For the toxic mushrooms, as far as ingesting them, obviously that's not good, but what about handling them?
Handling them is fine. You cannot get toxins on your hands from handling mushrooms that would be harmful to you even if you licked your fingers. So it's not something to worry about. Like anything you handle that comes from the ground, it has dirt on it, and I would always wash my hands before I cooked a meal or something like I would after I come in from the garden. But it's not something to really worry about. And please feel free to ask questions as we go along. I should have said that to begin with. Anybody else have questions at this point?
I have another question. Really
Yeah.
quick. As far as like the time frame of how long it takes, I'm sure all mushrooms are different, but maybe the Amanita, like to go from that little bulb to a full grown mushroom, how long is it?
It varies depending on the moisture and the temperature. And I don't know what else it varies on. I have this big spruce tree in my yard, and it has lots of different kinds of mushrooms that come up under it. They're all mycorrhizal. And so I've been watching these amanitas trying to come up. And for probably a week or more, they didn't do anything. They were just buttons. And then the buttons gradually grew, and then they were eaten by the deer. All I can say is and then other times I'll be in the woods one day and I'll see there's a button and I'll be back 3 days later and it's a mushroom. So I really don't know what makes the difference, because in my yard it was certainly, we've been having rain, the temperatures were moderate. I don't know why it took them so long. But something that's really fun to do with amanitas, and kids love it, is you take a button like that and you get some wet paper towel, put it in the wet paper towel and put it in a narrow bowl of some kind, and it'll grow. And you can watch it expand and grow. And it's really fun to see. I think it's neat, but kids go nuts for it. They just think it's wonderful. Okay, this is Agaricus augustus, and this sort of reminds you of the Amanita, and that's because it has this really prominent partial veil here, annulus. And Agaricus is, a favored group by people because we see a lot of them. They're not mycorrhizal, by the way. They are saprophytic. They're decayers. And a lot of them are edible, but some of them are definitely not edible. So this is a group that people are always interested in, but it can be tricky to identify them. And none of them are really poisonous, like you'll get kidney failure or anything. But as David Aurora says in his book, a number of them are part of the lose your lunch bunch. And Stropharia hornmanii has an interesting, we don't have this mushroom here today, it's kind of unusual, but it has an interesting remains of its annulus here. You can see there's some on the cap margin, and some mushrooms do have remnants of that veil on the cap margin, so that's something you also want to look for. And this is Cystoderma phalax, and it has this really interesting veil. It's like a big sock that it sits in. This is a Cortinarius, and Cortinarius has a cortina for a veil, and that's what all these cobwebby things are. The rusty brown spores have caught on the cobwebs, so you can really see them. Over here you can see they're just really beautiful, and we have some very nice examples of that today. And Cortinarius can also have a viscid veil, and come back. I went too ahead that time. All I have to do is let my little finger get over there and it moves. Cortinarius can also have a viscid veil, and viscid is this gooey wetness that you see here. There's my cursor, there it is. This gooey wetness that you see here, and we have examples of that also. Viscid is an important quality to recognize. And this is Foliota decora, and it has an annulus, but it's way up here. You have to really look for it. So you don't just only look farther down, you also have to look at the very top to see what's there. This is Galerina marginata, and it's an example of an annulus that is very friable. It doesn't necessarily, it's called evanescent, meaning it doesn't last there. So you need to find young specimens of these mushrooms and look at them to see if there is an annulus. This is the most toxic mushroom we have in Humboldt County. It has the same toxins as Amanita phalloides, the death cap, and I don't know how many of these it would take to kill you, but it isn't many.
One small one could do it a full-grown man, I believe.
One small one. And you wouldn't think, I mean, these mushrooms are maybe at the most 3 inches tall. You wouldn't think that anybody would be tempted to eat that. But people who are interested in psychedelic mushrooms are very at risk of confusing those with this. The difference is the spore color and the presence of the annulus. I don't know, Bradley, is there anything else I'm missing?
Yeah, well, the psilocybes are generally blue, the psychoactive ones stain blue when you bruise them, and this won't, but, you know, a crude appearance is similar. And they grow in similar habitats, too, in woody
They
debris.
grow on woody debris.
So, I don't hear about poisonings very often, but I worry.
Yeah, we do worry, and
Know
so
what you're picking.
I always try to make a point of that because it is something that is a risk. Okay, so now the hymenophore attachment, how the gills are attached to the stipe is another important, very important characteristic. So what I mean is, here are the gills, somewhere, where's my cursor? There. Okay, here are the gills, and they're attached here to the cap. Do you see that? They're not attached here to the stipe, they're attached here to the cap. So that is called free, meaning free of attachment to the stipe. And that's typical of Lepiotas, and this is Bulgariella, which is an unusual mushroom for us here. And Pluteus also has free gills, and here's a picture of it looking at it from underneath. And when you look at it from underneath, it leaves this real ring here, this vacant area at the very top of the stipe, because the gills are not attached to the stalk, they're attached to the cap. So that's free gills. Amanitas have what they call finely attached gills, and you can see that they're just barely attached to the stipe here. Hebeloma has bluntly attached gills. You can see that they just come right into the stipe at a 90-degree angle. This is a Tricholoma, and Tricholoma has notched gills. So right here you can see they come down, but they don't go right to the stipe. They do this little upturn. It's called notched, like a notch has been taken out of it. And that's typical of Tricholomas. And this is my favorite. This is, well, I like the little bug in there too, but these are decurrent gills. They run down the stipe. They are the absolute opposite of free gills. Free gills don't touch the stipe at all. These gills run all the way down the stipe. And we have a number of examples of this. These are also decurrent gills. Not quite as obvious as the other one, but they're decurrent. Okay, we'll talk about the shape of the cap. So this is convex. It's hard to read the writing on that thing, isn't it? Well, he calls it parabolic. I guess that is a better description. And this is, in the young ones, it's convex, and you may be able to just see that it's umbilicate. It has this little depression here. So he calls it convex to broadly convex and depressed. So that brings up something that's important, is that mushrooms change as they age. As we talked about, they start as a little button, and then as they get bigger, they expand. And the one that's second to the left, you can tell is young because the cap is still mostly rounded down. But as it gets older, you can see the one on the left is a little older, it's a little more expanded, and then the two on the right are fully mature. The cap is what they call plain, it's flat. And then some mushrooms in age actually become uplifted, where the cap is sort of turned inside out. I think I have a picture of that. This is Hygrophorus conicus, and it is an example of a conic shaped cap. Hygrophorus, not Hygrophorus. This is Mycena epipterygena, and it also is broadly conic. This is bell-shaped. This is Cortinarius rubicundulus. And it shows a yellow stain, if you notice that. And staining qualities are also important in identifying mushrooms. Some mushrooms stain when they're scratched or cut. Some boletes do that. Lactarius does that. Some russulas do it. They turn red and then black. So staining reactions are an important feature also. This is a conic cap. I love this picture.
Wow.
This is Cortinarius violaceus. It's the type species for the genus Cortinarius. And this picture shows the beautiful purple colors, and the one on the left, you can see the cortina. Beautiful mushroom, beautifully shaped cap. Okay, stipe attachment is also important. So this has a central stipe, and most mushrooms do. I mean, there can be variations because it's coming up under a branch or something, and so it doesn't grow right. But the stipe is centrally located. And that's a nice example of decurrent gills there on the right also. And then there are some that are eccentric, slightly off-center. This is Neolantinus kaufmannii, and look at how the edges of the gills are serrate. That's an important feature also. Hohenbuchelia petaloides has a lateral stipe. So does the oyster mushroom, Pleurotus striatus, has a lateral stipe. And those mushrooms are usually growing off of wood. Pinellus serotinus, I think we have a Pinellus over there, has a lateral stipe, and it's a pseudostipe. It's not really, see how short it is? There's not really much to it. And then this one has no stipe. It's just growing right off the wood, has no stipe at all.
That's the top we're looking at?
We're looking at the bottom.
Oh, the bottom.
It's growing off of something in the background there,
Oh,
probably a log. So it has no stipe. And then this mushroom starts out with a stipe that is central, and then as it grows, the stipe becomes absent, lateral and then absent. So that could certainly confuse you. I've never seen that mushroom. Okay, stipe consistency. Now this gets difficult. I'm not exactly sure how to explain this, but it has a lot to do with size. So big stipes are, what's the word I want? Fleshy fibers, thank you. And so these are fleshy fibrous, and it gets tricky when you get to little ones, which are called cartilaginous. So these are fleshy fibrous for sure, no doubt. Oops, let's go back to a fibrous one. The ones on the left are fibrous, are cartilaginous. And the only way I can really tell cartilaginous is to take the stipe and pull it apart, and if it comes apart in like strings, then that is cartilaginous. Can you think of a better way to explain that?
Well, the cartilaginous ones, Largen used to say if you have trouble, just go by size, and it's like,
Yeah,
I forget where the
that's what I
cutoff
do.
was, about 5 millimeters or something, call it cartilaginous, and above that, call it fleshy, but there's a texture thing. And when you're reading in print, it's hard to say what the definition of the person describing it is using, but the thin ones, the cartilaginous ones, usually are kind of brittle. And the fleshy ones, if you break it or pull on it, like you say, you'll see like stringiness to it. Fleshy, stringy stuff. But like you said, it's kind of a tough character.
It is.
They
a tough
certainly
character.
interblend, you know, and
The best thing is
One's
to
in
use
between.
size, I think.
yeah,
And
that's what Largen used to say.
in Aurora, he gives you the size, the diameter of the stipe as one of the characters, so you can use it that way. Okay. Some more fleshy fibers. So I end with this because I'd like to talk a little bit about collecting. If you're going to collect mushrooms, you want to be sure you don't put them in plastic bags. They die in plastic bags. So you want to use wax bags or even paper bags. Wax bags being best. You can get them at Murphy's, probably other places too. And you wanna use a basket or some kind of container that has vertical sides as opposed to a rounded bottom where everything falls together and breaks up. So something like this basket with vertical sides is good. Dr. Largent and I both use these Japanese gardener's knives. Sometimes you're trying to collect a mushroom in the forest and there's all these roots and twigs and things and it's hard to get at. It's very important when you're collecting a mushroom to identify it that you get the very base of the mushroom, because you wanna see if there's a vulva. You wanna see, is it spindle shaped, does it taper, what is the base of the stipe like? So it can help to have a tool like that knife. You can also use a little trowel, but you wanna remember that there's all kinds of mycelium there, and so you would prefer to disturb the mycelium that the mushroom's growing from as little as possible, although it's impossible to do that, of course, when you're digging up the mushroom. If you're just collecting mushrooms to eat and you know what the mushroom is, you don't need to identify it, and you can just cut it off, you don't have to dig it up. Let's see. Largent has a bunch of stuff. He's got clippers so you can clip branches out of the way. He's got a compass so you won't get lost. He's got a little hand lens so you can look for microscopic things. He's even got an altimeter, wow. And a whistle is a good idea. People do get lost. Yes?
If you cut them off, or, like take my kid, he likes to knock them over with a stick or something, does that actually hurt them, or can they regrow?
They won't regrow. It doesn't hurt the mycelium, but it could be that it prevents the spores from doing their job, which, you know, there's a reason that mushrooms exist, and the reason is to produce spores, which will eventually produce more mushrooms. So, you know, generally, I discourage kids from kicking over mushrooms. I explain to them what the mushroom is for, and they can often kind of get into that, actually. So, I don't know, that's the best answer I can give you, I guess.
Good enough.
People do kick them over, everywhere you go, you see they've been kicked over. When I pick a mushroom, there's a couple of things that I do. Now, Largent doesn't have it in his basket, but I have in mine a tool that I think is really great, and I'll share it with you. It's a mirror. It's a plumber's mirror, and you can bend over and you can put it under the cap of the mushroom, and you can see, does it have gills, does it have tubes, what, you know. And when you're beginning, you know, you may need to pick more mushrooms because you really need to see them. For me, and as you go along, you'll find that it may be enough to look at it with this and say, oh yeah, I see, that's a Swillis, I don't care about that. So you can get these for $9.99 at Ace Hardware. It's a plumber's mirror, and I really recommend that. But when you are picking mushrooms and you pick it up and you look at it and you say, oh, I see, this is a Swillis. Well, when you put it down, stick it back in its hole and put the duff in around it so you're disturbing the mycelium as little as possible. And the mushroom won't continue to grow, but the spores might drop. So if you put it in the right orientation, the spores are apt to fall and you won't have done much harm. So, this is the last slide, and it shows you the Wax Tex convenience bags that we use. And the other thing we use is these plastic tackle boxes. It's a great way to collect mushrooms, small mushrooms, although you can get some that have big, you know, compartments in them. But it's easy to carry, and it keeps them from, you know, getting knocked around and broken. So that's another really good way to collect mushrooms.
So that plastic won't harm.
No, actually, they keep pretty well in these plastic boxes. It's the plastic bags makes them slimy. I really can't tell you what the difference is. I mean, they're both plastic, maybe
I think it's
the
a
type
lack of
plastic.
air.
Lack
When
of
the-
air.
Yeah, 'cause they need to breathe, they're still metabolizing, and so, if they don't get any air, then they'll just die and rot fast.
They must get some air in these boxes
But
because
there's
they
air in the
last
boxes.
quite well in them.
Cause that holds the sides away from the fruit
Yeah,
body.
that's the other thing, the plastic bag, they're right up
They
against
just
the
collapse.
bag and they get slimy pretty quick. So,
And bacteria like them. And those little tubs, those are
Oh,
wonderful
yeah,
too, for the
the
bigger
little
ones.
tubs that you can get, you know, the little Glad ones with the lids, they come in packages of 4 or 6 or 8, and they come in all different shapes.
These kinds of
Virginia and I
little
have been using these.
I love these little tubs, and the things you get in the delicatessens, those little tubs. Keep those and just use them, because they really help protect the mushrooms from banging around. As she says, they're really fragile, and they just get damaged real easily. And I kind of pack them with dead leaves and stuff like that to keep them from banging against the sides.
And they're really inexpensive, and it's a good way to do it. You can use them over and over and over. So,
And leftover,
yeah, you know, the things you get from the
You get
delicatessen
you
with
get
food,
tomatoes in, you could use those.
You can recycle those by using those to collect mushrooms also. So, do you have questions? This is a good time to ask any you might have. Brendan, Scott, Bradley, Virginia, do you have anything you'd like to add to what we've talked about so far?
I don't believe so at the moment.
Yes?
Environmentally speaking, is there any species of mushroom that is unwelcomed? That
Unwelcome, you mean like
Like
invasive?
invasive.
invasive or that causes some sort of environmental, unwanted environmental effect?
Well, they're pathogenic.
Toxicity.
Run around
or-
your wall.
Yeah, there you go.
What is
The
that?
ones that grow out of your drywall.
Yeah.
Yes.
In your house, it's foundations.
Amanita verosa, yeah.
They're
I don't know if we have that here, do we? I've never seen it, Amanita verosa.
No.
I don't think we have it here.
They're pathogenic fungi, which foresters don't like, of course. Which is
Well, yeah,
Very much.
I mean, the pathogenic, the wood rotters like Armillaria mellia causes the root rot of the live oaks in Southern California. If you've ever lived in Southern California, you may have noticed they dig out these big circles around the live oaks to keep the moisture away from the roots to discourage the armillaria. But, you know, it's a natural process. It just seems bad to us because these are landscape plants that we like. But it is a natural process. It's just that we don't really like the results of it very much. Scott?
Well, I was just thinking if you wanted to consider ones that are unwanted, it's ones that came in from other countries.
Right.
And that's what I was really-
Foolishly, and then, they're pathogenic on the native species. And the things they came in with had defenses because they've been evolving with that pathogen for eons wherever they came from. But then we have a whole bunch of species here that are highly susceptible that have no defense. It's like the natives, when the white people came, they managed to win a lot of their battles because the natives got sick, right? So.
But are there fruiting body producing fungi that come in that category? I mean, I know
Well,
there's
the
a
one
lot
you-
of
the one you were describing
microscopic
earlier.
ones. Amanita phalloides.
The one that is not present in Humboldt that you were talking about.
Amanita phalloides.
That
Well,
particular
it's
one.
not, it's a pathogen for people who eat it, but I don't know
But I
that
was
it
thinking
has
for the
had
environment,
pathogenic
you know,
effects
what you were
otherwise.
talking about kind of gets to what I was asking about. Like, is there a negative side to its presence environmentally? And I think that kind of answered the question, what you said.
I don't know how many mushroom fruiting bodies, what Joanne was saying, how many that have mushroom fruiting bodies that actually harm the environment, like these microscopic ones do, these water.
Scott was
ones and
referring
whatnot,
to
but-
some of the pest
Sudden oak
diseases, like the one producing sudden oak death that are imported, but they're microscopic fungi. They're not the kind of fungi that we're talking about.
And they're actually not even fungi anymore.
Yeah,
No, they
they're
don't
They're
not.
more related
fungi.
to brown algae,
Yeah, they're
so
not
they're not
fungi.
included with
Yeah, but Dutch elm disease and.
Right, that's a
there's like
fungus.
White pine blister rust, those were
Blister
both
rust. Yeah.
introduced and
yeah,
Yeah.
chestnut
wiped out
blight.
trees, really wiped out a lot of trees, but it's part of the process, things. can
Yeah,
move
but
around
generally
the earth
they
and
don't have
change their
mushrooms.
distribution.
So
Amanita,
is mycology the study of just the fruiting bodied mushroom
No.
itself?
No.
No.
Mycology is the study of all fungi, from red molds all the way on up.
So it's that broad.
And so it's a very large and complex family, and I didn't go into this. Virginia does really great talk on ecology and biology of fungi. But fungi are not plants and they're not animals. They now have their own kingdom. They're recognized as an entirely separate group of organisms from plants and animals. But they do, their vertical scale goes all the way from bread molds to the higher fungi, which is what we're talking about here. We call them higher. I don't know if they are. Yes?
Could you talk a little bit about how one goes from newbie to kind of confident in the woods, and about how long it could take?
It's difficult, because for a lot of people it's difficult because of the nomenclature. If you're not familiar with Latin, it's more of a problem. I have a degree in biology and I was a microbiologist, so I was used to using Latin names for bacteria when I got interested in fungi. So I knew what the Latin names meant in English, and frequently they reflect characters of the mushrooms that help you remember them. So that is a problem for people at first. Another problem with learning mushrooms is there are just so many. And I really, today you're going to be exposed to a lot of different mushrooms just to show you the different features that we've seen. But when you actually go out and on a field trip, say, it's really good to just concentrate on learning a few. You say, okay, I'm going to learn what chanterelles look like today, and maybe I'll also learn russulas and lactarius, because those all have clear features. And just concentrate on learning what that genus is like. And if you take it slow, I know people who have learned fungi really well in a couple of seasons. So part of the problem is the mushroom season here is everywhere, is not very long. We start getting mushrooms if we've had rain in maybe late October, and then slowly it crescendos to mid to late November, and then it tapers off to where by mid to late January there's not much left. So if you want to learn mushrooms, you have to do it rather intensely during that period, because it's not like they're here all year and you can work on them. So if you really want to learn them, you have to apply yourself pretty intensely during that period. And we have field trips twice a month. Bradley leads one on the Saturday before our meeting, which is the third Wednesday, and I lead one on one Sunday a month, and it varies according to my grandson's soccer schedule and other things. And that's a really good way to learn. Bradley is really good about explaining and teaching on his trips, and my trips are always to this one place in McKinleyville called the Van Eck Forest, where we have permission to go and to do a survey and make a record of the fungi we find. And we're now, I think, in our 5th year of doing that. And the purpose of my trips are strictly educational. If you want to go looking for edible mushrooms, don't come on those, because we may find a few and you're welcome to take them, but that's not the purpose. The purpose is really to teach you how to identify fungi. So it's we usually have fairly small groups on those field trips, but they're kind of fun. So we have 2 field trips a month. Another thing you can do if you want to hasten your learning prospects is you can go out with people in the club who go out informally. And there are a number of people who do that. And you can call them up and say, hey, are you going to be going out? I'd like to go with you. Mike is a person who does that. I'm willing to do that too. And I think it would be really nice maybe if in our newsletter or by email we had a better way for people to get in touch and go out on their own. And really a good way to do it, which we haven't done in a while, is to get small groups of people together with mushrooms they've collected to identify them and key them out. And I did that a few years ago with a small group of people who came to my house every Wednesday night, and every one of them really learned their fungi pretty well. I'd say by the end of the 2nd year we did that, they were doing really very well, because working one-on-one or in small groups with people, as we're going to do today, is a really good way. You go out and collect fungi, then you have a time that you meet and you key them out, and we'll do a little bit of introduction to keying here a little bit later. So I don't know if that answered your question. I hope it did. It isn't something that happens really fast.
It's true, isn't it, that mushrooms do not hybridize?
You know, I think that there's debate about that, because there are some indications that in fact they do. Scott's gonna speak to that, I think.
Well, no, I'm not gonna speak prophetically to that or anything, but it's the reason that we all had our little is because for a long time it was drone that it's great, this species has been around for 1 million years and it's the same one that they've had over in Europe, etc., or whatever, you know. And then they're finding out now that they are not the same species, so that's one thing. But that also, a lot of the fungi change their appearance over time in the woods, so they've been classified as different species. And in terms of actually hybridizing, I think they're starting to find out that there are fungi that are integrating just like plants do. They're a little bit more dynamic that way than was thought. So it's tricky. And that could be a problem with introducing one of these other non-edibles. If they do end up finding out they like mating and they're successful, then you might end up with a mushroom that is no longer edible, but it looks everything like your edible one.
But that hasn't happened yet.
Not hasn't, but you can't say it won't happen. Never.
Probably not in our lifetimes, though. One of the things that you should know about this time at which you're entering into an interest in fungi is that DNA sequencing is changing mycology, just like it's changing plants and everything. And so names are changing, and many things are being named all the time. I don't know if you're aware of this, but there are, I'm not good with remembering numbers. I can't remember the numbers of how many species of fungi they think there are, but just a fraction of them have ever been described and named. So it's a great area for citizen scientists, because now that we have digital photography and we have websites like Mushroom Observer, people are, you know, posting pictures and sending specimens for sequencing, and all of a sudden the number of mushroom species that have names is increasing very rapidly. So it's kind of an exciting time, and it's also a little overwhelming. And I have a friend who is very involved in all this, and he told me that what I should do is just stick with the names that I learned, and don't worry too much about the new ones, because they're probably going to change again. So, you know, just don't worry too much about changing names. If you have mushrooms demystified and you're working from that, just learn that, and we'll deal with all these changes as they come along. Okay, yes.
And since Joanne mentioned it, another thing to address your question about how to learn them is to put yourself out there. Keep going out and collecting the mushrooms and photographing the mushrooms. Work with the keys. Try to come up with a name for it. Post your photos on Mushroom Observer, and just be willing to accept the fact that whether you got the name right or not, there are going to be others who are going to weigh in and offer you their name for it. And if you just really didn't have a name for it, it's a great way to get a name for it.
Yeah.
Because that's what happens at the mushroom fair is we make these great collections, and even those of us that know a few species, we make this great collection, and we're just really excited, right? I'm speaking we, even though it's me, and I know you guys agree, right? But you don't know what it is, and then it gets a name from somebody that did know what it was. So it's a pretty sweet way to go about doing it. Just keep collecting them until, you know, or at least photograph it.
And Mushroom Observer is a good website for that. I like it. I think it's really useful. And don't worry about if you don't know what it is. Just put it out there and see what kind of response you get. All right. The next thing we're going to do is take a short break. And I'm sorry.
I'm just curious. Are there any mushrooms that you don't want to pick or disturb, like knowing that they might be like endangered or extremely rare or anything?
Not that I can think of.
Okay.
I mean, not that you would know.
Yeah, it's hard.
Scott, do you wanna
If you
comment?
see monumentally large fungi in the woods, some of those species only ever grow with really large old host trees. And so, since there are less large old host trees
Best
out
to
there,
leave them
then the numbers of those types of fungi are diminishing. And so that's something I would avoid messing with. There's not much you can do with a large super mondo fungus except display it anyway, generally.
Yes.
Does southern Humboldt have a drastically different type of fungus than northern Humboldt?
Well, the type of fungus you have depends on the type of trees you have. If you're talking about mycorrhizal fungi, which is what we mostly are. So the trees that grow with tan oaks in northern Humboldt are pretty much the same as the trees that grow with tan oaks in southern Humboldt, and the same is true with the redwood forest and Doug fir. I think they're fairly similar.
But if you were at an area where there's a lot of old growth compared to some place, like say the community forest where there's not a whole lot, will you see different things?
Well, if you're talking about redwoods, the
Yeah.
answer is no, because redwoods do not have ectomycorrhizal associates. So they do have mycorrhizae, but they're not the kind of mycorrhizae that produce big fruiting bodies. So the mushrooms you find in redwood forest, if it's a pure redwood forest, those mushrooms are saprophytes. They're not mycorrhizal. If there's Doug fir mixed in there or some spruce mixed in there, then you will have mycorrhizal fungi. But if it's just redwoods, they're decomposers. So I would expect it would,
But
be
there's-
pretty much the same.
There's some
So-
specialized fungi on redwoods, though, aren't there? Decomposers.
I
aren't
thought
they?
that
Like redwood
redwoods
needles.
have endomycorrhizae, and I thought they
No, I
did
mean
not produce fruiting bodies.
I mean the decomposers.
That's true, that's what I've heard.
Well, there'll be decomposers in the redwood forest like crazy. There's lots of them, because
Which might be
there's lots to
specialized,
decompose in the
redwood-
redwood forest,
redwood
but
wood
not
and-
mycorrhizal fungi.
There may be some other associations, like you were saying with the hygroscides, from what Ilse was saying. They
Right.
found some really interesting associations that
They're finding now that it isn't just trees, that there are other vascular plants that have mycorrhizal associates, including possibly grasses, which is interesting, but kind of outside the scope of.
But they're not
what we're talking-
ectomycorrhizae.
No, they're
and
endo.
But they are mushrooms,
Right.
so who knows?
There's a lot that's not known about mushrooms. I'm
Even
sorry I can't
ecology
give you
is
definitive
getting
answers
real
to all your
complex.
questions. Number one, I don't know all that much, and number two, nobody knows the answers to some of these questions. It's really, a fairly unexplored kingdom compared to plants and animals. So there
They're harder
aren't
to
a lot
study.
of answers to things.
Lots of stuff going on underground and it's really hard.
So are there any more questions? Yeah.
You mentioned something about the growing season for wild mushrooms, October through January. Okay, as far as mushrooms in the garden, growing mushrooms in the garden, is that under the same restrictions? Or
Are you talking about cultivating mushrooms?
cultivating, right?
That would depend on the type of mushroom and what its needs are. So I can't speak to that. You would
Whether
have to
you're watering
read.
it.
And
Yeah,
if
whether
you're
you're
watering it,
watering it.
of course. But you would have to read the literature that comes with, like Stropharia rugosa annulata is one that I think that they sell. And I don't know when it fruits. And I didn't mean to say.
It'll fruit in the spring and summer, very unlike our local mushrooms.
Yeah.
And it does well here in the garden.
Yeah, I've had it in the garden.
You've had it too, yeah.
But it peters out after a while,
Yeah,
but
it doesn't last.
it decomposes your mulch in your garden. And so it seems to me it did fruit in the spring and summer when I had it. But that would be completely, I mean, that's not a native mushroom here. It's not native to our area, so its whole thing is different. But I didn't mean to imply that we don't have mushrooms at other times. We do, but it isn't like it is right now, where they're just everywhere. They're rare. And then there are mushrooms that fruit in the spring, especially in the mountains where their snow is melting. That's where you find the morels, which you've probably heard about. And we do have morels in the mountains here, especially in areas that have burned recently. Up in Oregon, where we have a cabin, people look for them in areas that have been logged. They like disturbed ground. There's a whole host of fungi that are found in snowmelt areas and in Mushrooms Demystified. He has a section where he talks about different habitats and what mushrooms you find there. So he has a section on snowmelt fungi, he has a section on burn fungi, he has a section on fungi that grow in grass, yada yada. And that's kind of fun because there's not that many snowmelt fungi. And so it's fairly easy to learn them because you're not overwhelmed with all these choices. So you can go out in the spring and find fungi. If you go into the mountains, I know in southern Oregon, there are fungi that fruit there in the spring. They have summer boletes there that everybody collects. So I don't know if that's true in our mountains here, but I think it's possible.
By Shasta, they get.
At Shasta.
And in our mountains, we get summer fungi, a little.
Little.
It's nothing like the fall, though.
Yeah, so I mean, it's not like there aren't fungi any other time. There are, but they're not that many. So learning them ends up being very compressed. Every year as we come into the fall, I get real anxious about how am I going to do all the mushroom stuff and do anything else, because there's so many mushrooms, all you want to do is mushrooms.
Which also, in learning your mushrooms, they come in a certain sequence, okay, a certain chronology they appear. So if you're wanting to learn your mushrooms, one of the ways you can take it and break it down into little chunks along the way is start early, real early, and get your head into your mushroom books early with the early mushrooms and get those out of the way so that 2 and 4 and 6 weeks later, even if those are still out there, okay, you kind of got those kind of done sort of for now, you know, and then you can work more readily on the plethora, because when the horde comes, it's there, and you know, you're just going to have an endless bunch. But if you can kind of have, say, 20 of them or whatever, you're just like, oh, that's the blah 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 again, then you're done with that. So you can do that for the early ones and all the conchs you see all the time. You try to get those in your head when there's nothing else out there, and it really improves things further into the season, because then it's crazy.
So are you noticing climate change affecting the season that you're talking about?
You know, it's hard to say. I mean, certainly this season has been very different. The rain started early, like it used to. Back in the 70s, we got rain in late September, and our mushroom fair used to be held around November 7th or so, originally. And then we just had to keep moving it back because the rains were coming later and the mushrooms were coming up later, and now we're the weekend before Thanksgiving. So we're about 2 weeks later with the peak of our mushroom season, I would say. This year has just been really strange because it has remained so warm, and usually the cold weather brings an end to the boletes and a lot of the mushrooms that we're still seeing, but they're still coming up. So I don't know. I know that the data shows that climate change is real, and I believe that. I accept that. But whether we can say that what's happening now with fungi is related to that, I don't know. There was a study that was done 30 years ago by a woman named Sue Van Hook, who was a graduate student at Humboldt State, and she did this really great master's project on the mushrooms of Lanfear Dunes. And I'm very interested in doing a follow-up study on that to see if the time of fruiting and the number of fruiting bodies and the species of mushrooms fruiting has changed significantly. She did an extensive study relating the rainfall to the time of fruiting, and that was back in the day when mushrooms did fruit earlier than they do now. So I think it would be very interesting to do that study, but it's complicated to get the permits and everything to do it, so it hasn't happened yet. But that's the kind of thing that would be interesting, because a 30-year period is fairly significant, so if you did this for 2 or 3 years to try to duplicate her results and compare it, it could be very interesting. Okay, let's take a short break, and if you would help us move the chairs, well, let's see, we have to bring round